Hey there, science fans. Have you ever thought about the tiny things that can cause big trouble? I'm talking about viruses. They're everywhere, from the air we breathe to the surfaces we touch. Viruses are incredibly diverse, coming in all shapes and sizes. Some are shaped like spiky balls, while others look like tiny spaceships. And don't let their size fool you. These microscopic invaders can cause a wide range of diseases. But how do scientists keep track of all these different viruses? That's where the Baltimore classification system comes in. This clever system, named after virologist David Baltimore, groups viruses based on how they make copies of themselves. Think of it like sorting your socks. You might group them by color, size, or purpose. With viruses, we group them by their genetic material and how they replicate. Pretty neat, huh? The Baltimore system is like a virus filing cabinet, helping scientists organize and understand these tiny troublemakers. Imagine trying to sort through a chaotic pile of papers without any system in place. The Baltimore system brings order to the chaos, making it easier for researchers to study and combat viruses. It divides viruses into 7 groups, each with a unique way of replicating. This classification is crucial because it allows scientists to predict how a virus will behave and spread. These groups are based on the type of genetic material the virus carries, either DNA or RNA, and how it uses that material to make more viruses. DNA and RNA are like the blueprints of life, and understanding these blueprints helps scientists decode the virus's replication strategy. Why is this important? Well, understanding how a virus replicates helps us develop ways to fight it. By knowing the replication process, scientists can identify weak points in the virus's life cycle. For example, knowing that a virus uses RNA to replicate allows scientists to design antiviral drugs that specifically target that process. This targeted approach increases the chances of successfully neutralizing the virus. It's like having a secret weapon against the enemy. With this knowledge, scientists can develop more effective treatments and vaccines. But before we dive into the nitty-gritty of each group, let's meet a few viral families and the diseases they cause. Understanding these families helps us grasp the broader impact of viruses on human health. We'll explore the Adenoviridae family, known for causing the common cold. These viruses are widespread and often lead to respiratory infections. The Herpesviridae family, responsible for cold sores and chickenpox, these viruses can remain dormant in the body and reactivate later. And the Orthomyxoviridae family, which includes the infamous influenza virus. This family is notorious for causing seasonal flu outbreaks and pandemics, making it a significant focus of research and public health efforts.
First up, we have the Adenoviridae family. These viruses are masters of disguise, often causing respiratory infections like the common cold. They are known for their ability to evade the immune system, making them particularly tricky to deal with. They can also lead to conjunctivitis, more commonly known as pink eye. This condition can be quite uncomfortable and is highly contagious. Adenoviruses are highly contagious, spreading through coughs, sneezes, and contaminated surfaces. It's important to maintain good hygiene to prevent their spread.
Next, we have the Herpesviridae family. These viruses are notorious for causing lifelong infections. Once they enter the body, they can remain dormant for years. They include the herpes simplex virus, responsible for cold sores and genital herpes, which can be painful and recurrent, and the varicella zoster virus, which causes chickenpox and shingles. Shingles can be particularly severe in older adults. Herpes viruses are sneaky, hiding out in the body and reactivating during times of stress or weakened immunity. This reactivation can lead to outbreaks and further complications.
Finally, we have the Orthomyxoviridae family, home to the infamous influenza virus. Influenza, or the flu, is a respiratory illness that causes fever, cough, and body aches. It can be debilitating and often requires bed rest. It spreads rapidly through respiratory droplets and can lead to serious complications, especially in young children and older adults. Vaccination is key to preventing severe cases. These are just a few examples of the many virus families out there. Each family has its own unique characteristics and challenges. Now, let's explore how the Baltimore system classifies these tiny troublemakers. This system helps scientists understand and categorize viruses based on their genetic material and replication methods.
Group 1 viruses are the most straightforward of the bunch. They carry double-stranded DNA just like our own cells. This means they can directly use the host cell's machinery to replicate. Think of it like walking into a factory and using the existing equipment to build something new. Examples of Group 1 viruses include the aforementioned Adenoviridae family, as well as the Papillomaviridae family, which causes warts and some types of cancer. These viruses are generally stable and less prone to mutations than RNA viruses. Section 5. Group 2. Single-stranded DNA viruses. Group 2. Viruses are a bit trickier. They carry single-stranded DNA which needs to be converted into double-stranded DNA before it can be used to make new viruses. Think of it like having a recipe written on a single sheet of paper. You need to copy it onto another sheet before you can start cooking. One example of a Group 2 virus is the Parvoviridae family, which causes a range of diseases in animals, including parvovirus in dogs. These viruses are generally small and simple with limited genetic information. Section 6. Group 3. Double-stranded RNA viruses. Group 3 viruses are unique because they carry double-stranded RNA. This type of genetic material is not found in our cells, so these viruses need to bring their own special enzymes to replicate. It's like showing up at a construction site with your own set of tools. One example of a Group 3 virus is the Herpesviridae family, which includes rotavirus, a common cause of severe diarrhea in infants and young children. These viruses are often resistant to environmental conditions and can survive for long periods outside of a host. Section 7. Group 4. Positive sense single-stranded RNA viruses. Group 4 viruses carry single-stranded RNA that can be directly translated into proteins by the host cell. Think of it like having a set of instructions written in a language the cell already understands. This group includes a wide variety of viruses, including the Picornaviridae family, which includes poliovirus and rhinoviruses, another common cold culprit. Group 4 viruses are often responsible for outbreaks due to their high mutation rates and efficient replication strategies. Section 8, Group 5, Negative sense single-stranded RNA viruses. Group 5 viruses also carry single-stranded RNA, but it's the opposite of Group 4. Their RNA needs to be converted into a positive sense strand before it can be translated into proteins. Think of it like having a set of instructions written in a mirror. You need to flip them around before you can read them. This group includes the Orthomyxoviridae family, like flu viruses, as well as the Rhabdoviridae family, which includes the rabies virus. Group 5 viruses are known for their ability to cause severe diseases and pandemics. Section 9. Group 6. Reverse transcribing RNA viruses. Group 6 viruses are real rebels. They carry single-stranded RNA but use a special enzyme called reverse transcriptase to convert their RNA into DNA. This DNA then integrates into the host cell's genome, becoming a permanent part of its genetic blueprint. The most famous example of a Group 6 virus is the Retroviridae family, which includes HIV, the virus that causes AIDS. These viruses are particularly challenging to treat because they become a permanent part of the host cell's DNA. Section 10. Group 7. Reverse transcribing DNA viruses. Group 7 viruses are a bit of a hybrid. They carry double-stranded DNA but use reverse transcriptase at one stage of their replication cycle. Think of it like having a blueprint that needs to be temporarily translated into a different format before it can be used to build something new. One example of a Group 7 virus is the Adenoviridae family, which includes hepatitis B virus. These viruses can cause chronic liver infections and are a major cause of liver cancer worldwide. Section 11. Why classification matters. Understanding, treating, and preventing viral diseases. In this section, we delve into the significance of virus classification and its impact on our ability to combat viral diseases effectively. So there you have it. The Baltimore classification system in a nutshell. This system categorizes viruses based on their replication strategies and genetic material, providing a framework for understanding their behavior. But why is all this classification so important? It's not just about naming and organizing, it's about gaining insights that are crucial for medical science. Well, understanding how viruses are related and how they replicate helps us in countless ways. It allows researchers to draw parallels between different viruses and predict their behavior.
First, it allows us to develop effective antiviral drugs and vaccines. By knowing the specific characteristics of a virus, scientists can design targeted treatments that are more likely to succeed. By targeting specific steps in a virus's replication cycle, we can stop it in its tracks. This precision is key to creating treatments that are both effective and safe.
Second, classification aids in disease surveillance and outbreak response. It helps public health officials track the spread of viruses and respond swiftly. By quickly identifying the type of virus causing an outbreak, we can implement appropriate control measures to prevent its spread. This rapid response is crucial in containing diseases.
Finally, understanding virus evolution helps us predict and prepare for future outbreaks. By studying the genetic changes in viruses, we can foresee potential threats. By studying how viruses change over time, we can anticipate potential threats and develop strategies to mitigate their impact. This foresight is essential for global health security. The world of viruses is vast and constantly evolving. Each discovery brings us closer to understanding these complex organisms. But with systems like the Baltimore classification, we're one step ahead in the fight against these tiny troublemakers. This system is a powerful tool in our scientific arsenal. Keep asking questions, stay curious, and remember, science rules. Your curiosity and dedication to learning are what drives scientific progress.